So who we have here, this is Dr. Jerome Maller, and I just will go back a bit. So about 11 years ago, this is back in the days when I was based at Stanford, we were, it must have actually been 6 years, more like 7 or 8 years ago, Jerome came to Stanford, and immediately we got talking, bouncing off ideas off each other. And it's sort of ever since then we've had this wonderful sort of korero with Jerome and myself, and this relationship that grew into a relationship with Mātai and GE Healthcare. And so, in the last couple of years, we've, as you know, many of you know, we've bought a wonderful GE system. It's a GE 3 Tesla MRI system. We call it a Lamborghini of imaging. And now we've got Dania, who's joined GE, must have been a year ago, who's our research relationship manager, who is so excited to host in Gisborne for the first time. It's been just a real wonderful partnership we've got. So the other person I want to mention is Hari, our on-site scientist. Where's Hari? Hari right over there, and his son Yogesh there. Hari is our on-site GE scientist that helps to keep us up to date with the technology. He's got direct links into Jerome and Dania and into headquarters up in Milwaukee in the USA. So it's just a wonderful relationship. They keep us at the cutting edge in the imaging space, in the brain imaging and the body imaging space, and we're very excited to have them both here. So I was just going to introduce Jerome. So Jerome, his little bio on the web is probably about 2 lines, and it's the most modest bio I think I've ever seen for someone who's achieved so much in his career. I think Jerome's done everything that a scientist in our field would ever have dreamed of doing and accomplishing. So he's an MRI clinical science specialist at GE Healthcare. He's a neuroscientist and an adjunct associate professor at Monash University. He's an adjunct professor at the Australian National University and a research fellow at the University of Sydney. His focus is on multimodal integration of brain measurements and simulation technologies. And in terms of his expertise, I mean, I'm holistically, there's MRI, there's traumatic brain injury, there's something called diffusion tensor imaging, there's depressive disorder, there's neuroimaging, there's image processing, and he's also written a handbook of structural brain MRI analyses, which is very, very well known. So it guides a lot of us, people like me and the Mātai team. And he's got 150 publications that have been cited over 7,000 times. And he's also done research into brain modulation, so using things like transmagnetic stimulation to change the way the brain thinks and things. And I haven't explained that very well, I'm sorry, Jerome. So when Jerome, when we first got the machine, the MRI machine, Jerome was the one that was zooming into the modular, because we've got a little device that he can zoom in and help us drive the machine and guide us into the latest technology. He's also mentored our PhD students. We've got Mary here. Where's Mary? Over there. He's mentored Mary, who's now a research fellow at Mātai, and we're collaborating on several projects, including ADHD, concussion, and also on our internship training program. He's taught into our training program. And so he's a wonderful scientist. Sorry I'm bigging you up here, Jerome. I know you're a modest person, but you're really a wonderful scientist and a great teacher, and I don't actually know how he actually sleeps. I don't think he sleeps. Big welcome to Jerome, and I'll let you take over, Jerome.
Thanks. Kia ora. Thank you for inviting me to come. First, I want to say Dr. Dudley's presentation was magnificent. In 25 years of neuroscience research, I've heard a lot of presentations. And to present something as complex as dementia is not easy in 1 hour. To do it succinctly in language that everyone can understand is a very difficult task. So kudos for that. The reason I really loved it as well is because before I did my PhD 20 years ago, I was a child neuropsychologist. So everything was about strengths and weaknesses, how to assess someone in aspects when they didn't even realize what you're actually trying to assess. That's a real skill. And some of the examples that Dr. Dudley presented were very clever. Some of them were basically taken, or not taken, they were modified from existing techniques in a very clever way. And it's very hard to do that in a culturally specific context. So this new tool, from what I can see, is extremely unique. I can see it being very helpful. She also touched on some key terms which I could probably talk about for a day, just on each term and how relevant they are, such as she mentioned this term cognitive reserve. I've been looking at that for over 20 years, and there are thousands of publications showing how important something like cognitive reserve is. So we all have cognitive ability, and the more education, obviously, the more connections you have some reserve before you start showing symptoms of dementia. That's really what a lot of the research is trying to show by the term cognitive reserve. But again, I could talk all day about that. And all these other terms that she touched on were right on the money. Each term is really important when we're talking about aging and conversion in some people to dementia. One thing to add, a really important question someone asked was the difference in the incidence of males versus females. That does change from country to country. So Japan's a really nice example where it actually does change from generation to generation because they think of the different like or dislike of raw fish. So talking about omegas and vitamin D and vitamin E and how that differs around the world. But we also have to bring in, unfortunately, genetics. So there are a few genes known to directly relate to the onset of dementia. And I won't go into that, but suffice to say there are some major ones and they're referred to as ApoE. So it's up to you if you want to research some of that. The real question about that is, do you really want to know? Because there's nothing you can do about it. So we call those alleles. And we give you two numbers, 44, 24, 22. And we say, right, if you've got this, there's a higher probability that you may eventually develop one of the many disorders of dementia. The other point was, as she rightly pointed out, Dr. Dudley said, Alzheimer's is not the only dementia. There's at least 20 types of dementia. And with each type of dementia, there's at least another 20 types of type of dementia. So the major one we talk about is Alzheimer's disease. There's at least 20 types of pre-Alzheimer's. The most common, which Dr. Dudley alluded to, is called MCI, mild cognitive impairment. But there's also AMI, age-associated memory impairment, and it goes on and on and on. So this broad term dementia doesn't really mean that much. It's just a diagnosis. To break it down is more important. Someone mentioned that their relative maybe had some more aggressive tendencies. Well, that could actually represent a different type of dementia. Vascular dementia, frontotemporal, so more emotional. Lewy body dementia can have more of those tendencies as well. It's very important in the treatment regime that is established, rather than just saying, unfortunately, a relative has dementia. We need more than that. But I'm going to talk about psychiatric illness using three major or more common illnesses. So dementia, schizophrenia, and depression. And again, in each of those streams, there's not just schizophrenia, there's all types of schizophrenia, with or without auditory hallucinations, with or without visual hallucinations, psychosis, it goes on and on. When we're talking about depression, is it major depression? Is it treatment-resistant depression? Are we incorrectly talking about bipolar 1 or 2? There's all these different breakdowns, and unfortunately they're guided by the psychiatric holy grail, the DSM, Diagnostic and Statistical Manual, which really doesn't help us that much. It's not very useful in the end, and the criteria keep changing from edition to edition. 1 year autism's in it, the next one it's not. How do you define these things? So I'm going to try and speak about some of these things as simply as I can. If you try and break down something complex too easily, you are trivializing it. You're not really giving it the complexity that it deserves. So I always try and explain these things as simply as I can, but not to such a level that it just seems trivial and it's like, so what? So the first thing I'm going to show is a picture of the very first MRI ever acquired and published. 3rd of July 1977. Most MRI nerds like me know that date and they know this picture. So believe it or not, this is an MRI. This is the inventor's student who doesn't look too happy because no one knew what was actually going to happen. There's a long story behind it. Suffice to say, there were nurses standing there were physicists standing there were paramedics standing there saying, this person's not going to survive. You're putting through all this magnetism. None of that happened. None of that happened at all. This is a big magnet. There's a whole lot of copper near it. This is an antenna. You get one image right through the middle. And after a day or so of all the computers processing, this is what they got. A slice going through. Unfortunately, the person who did this didn't get the Nobel Prize, which is very controversial. Two other people did, but I won't go into that. But he did pass away last year, which is still very sad. So this is where we started. This is where we are now. So this is, and I am from GE, but my talk is not about GE. My talk is about psychiatric illness in the context of neuroscience and neuroimaging. But suffice to say, we started on the previous slide and now we're at this. So this is the same scanner that Marta utilises. So it looks very different. The fundamental principles are very similar, except now we don't just sit there and get one slice. We lie down and we can get the whole body. And it's a lot faster. It doesn't take 24 hours to get an image. It happens instantaneously. So we can scan the body and say we do a slice here, we call it a slice. We can reconstruct up to 60,000 slices per second, which is really quite amazing. This is where computer technology is at the moment. This is what the antenna looks like, for example, for the head. Just put your head in and that's an antenna. This is a blanket. It looks like a blanket, but it has a whole lot of antennae in it. You just put it on the person and plug it into the table. This is the sort of level of technology we're up to now. But really, today I'm going to be talking about this sort of technology with this sort of antenna. What can we get from that? So the first thing to understand is that when we look at an MRI, has anyone ever had an MRI scan before? Yep. So you know there's a lot of banging going on. Yep. Because when electricity is put through copper coils, they slightly expand. That's how a speaker works. That's why you get sound out of speakers. It's the copper expanding and contracting. To do that, we're playing with some of the water molecules. You don't feel it. You wouldn't feel it on any level. But basically the water molecules are giving off a signal. If something spins like this, if something spins 5 times per second, and then you match it with a frequency that says, I'm going to spin you 5 times per second, but a lot of power, you're going to make it move. And as it comes back, the antenna listens. Ooh, I hear it. I hear it. And that's, again, not to dumb it down too much, that is how we get this sort of signal. The antenna's listening to it. And what we end up is with an image, but it's not like a digital camera. We're trying to go inside the brain and we take this digital image. This picture here, which is sort of going through my ears, is made up of about 200 rows and 200 columns, which is really quite remarkable. And that only takes a second, sometimes a fraction of a second to acquire, and then we do it through the whole brain. So if we magnify it, we can see that we're actually getting squares. But in a digital camera, you've heard these terms, megapixel. How many megapixels is your camera? How many of these squares can we get in one image? Well, MRI is 3D. So it's not just a picture. This screen has a depth. So we don't say pixel, which is a picture element. We're now getting a volume element. So we call it a voxel, not a pixel. So when we acquire the MRI, we're getting voxels. If I, on the computer, circle this, I say how many voxels are there? There might be 100 voxels. and each voxel is 1 millimetre cubed. The size of that is 200 millimetres cubed. That's the essence of how MRI measures things. That's the absolute basics. So I'm going to try and put all this in the context of dementia, schizophrenia and depression, and how the size of these things in different parts of the brain relate to these disorders. So as was very accurately said, is that we have this overarching term dementia. The most common form is Alzheimer's, but by no means is it the be-all and end-all, because we have all these other types that have just been mentioned. So it used to be, even for my PhD over 20 years ago, I was interested in this thing called the hippocampus. So that picture you saw of that slice, I measured, say, 50 slices, slice by slice, and then multiplied that by how many voxels are there. Well, there's 2,300 voxels. It just took me an hour to do. So 2,300 multiplied by 1 is 2,300 millimetres cubed. And you put it in a database. Forget all that now. The computer does it all for you, and it does it in every part of the brain, and it does it in a matter of hours like that. And it's actually clinically validated as well. There are many neurologists who pay for services to send their data to a company. It comes back an hour later, everything's automatically done for them. It's really quite beautiful. But it was validated against people like me, unfortunately, who had already done it manually thousands of times. Then they refine their mathematics and go, oh, we can do this like this. So now we can start looking at thousands of scans all at once and get the results back the next day. Instead of taking, like me, an idiot, took 3 years. Bang, you just get it done now. So the most common sort of dementia is Alzheimer's disease. And really, what's the first thing to look at on a brain scan? Lucky me, hippocampus. So at least I was looking at the right area, considering my thesis was on preclinical dementia. Okay, so this is a normal scan, or someone who doesn't have anything detected. Their memory seems to be age appropriate. Their personality seems to be, as you'd expect for a normal, healthy person. And I'm circling this thing here called the hippocampus, which is a real buzzword, but there's a reason why it's such a buzzword. So hippocampus in Latin, although some people may argue, it translates to seahorse. Because in 3 dimensions, as I'll show you, it does sort of look like a seahorse. It has a big head, and it curves around, and then it has a tail which goes on an angle. So we're looking here, all the black is liquid. So if our head size doesn't change from about the age of 7 until we're passed away, the head size doesn't change. And if the brain is set at roughly the age of 25, then if there's less brain, there must be more liquid. Or if there's more liquid, more what shows up as black, there must be less brain. So a lot of us don't actually look at the size of something, we look at the size of what is not there. And what is not there is liquid. There's liquid there, so there's less brain. There's less brain, there's more liquid. So if you focus on that, there's a little bit of black there, which is liquid. And now I show you a person of the same age who has diagnosed Alzheimer's. There's more what's showing up as black, which is liquid. If there's more liquid, there must be less brain because the size of the head hasn't changed. So this is clear-cut Alzheimer's disease. But it's not the be-all and end-all, because Alzheimer's doesn't come in and go, oh, I'm just going to target one area, you hippocampus, I don't like you. No. It does start at least 2 places at the same time by these terms you might have heard of called plaques and tangles. But I won't go into plaques and tangles. What it does do is it says, okay, these areas are more vulnerable. They're going to get smaller to start with. But at the same time, a generalized reduction, we call atrophy, will also happen. We don't need to measure these things when it's obvious. As Dr. Dudley correctly showed, you might have sort of missed it on that first slide, but it's these gaps in between brain structures. This black, which is liquid. In reality, by the way, it's actually clear. It's just on MRI it shows as black, but in reality it is clear. So this is the size of this area between two brain structures, and now look how big they are, and the middle bits, how much larger they are. It's not just hippocampus, but it's a giveaway that this brain is not healthy. But we don't just see this in Alzheimer's, we see it in other disorders. Medication-resistant, long-term, chronic, untreated major depression. But here's, just back on Alzheimer's, here's the same person. 1.5 years time difference. So the scan's the same person. Here they were at baseline, 1.5 years later, and again 1.5 years later. The hippocampus of all structures has gone significantly smaller. You don't need to measure it, you can see it. But you can also see the liquid, the spaces, have gotten larger. So this is advanced Alzheimer's. Not everyone's, by the way, changes so quickly. This is just an example from the literature of an example of someone who was dementing quite quickly. So what does it look like from different perspectives? This is from in front. You have one on each side. On the side. And this is from on top. So you can see very roughly, it's got this seahorse shape. I'll show you more of the seahorse later. It might not be so clear here. But another thing I'll point out is that if this is what we call the hippocampus, there's a ball that sits on top of it and in front of it called the amygdala. And this is emotional memory. And this is relevant to all psychiatric illness because that sometimes doesn't get smaller, it gets bigger. There's a famous article, 2001, and it says, bigger is not better. The person was absolutely right. Because what was happening between brain cells in some people is there's edema, there's inflammation in between brain cells. But we can't pick that up on weak scanners. We just get this gross picture and we go, oh, it's bigger, must be better. No, size is not everything. And in the brain, bigger is not necessarily better. It has to do with efficiency. So in the beginning, we'd take two brain scans, the same person, 1 year apart. We'd put one over the other and say to the computer, tell us in colour where the difference is. And we can see the difference in this person who is suspected of Alzheimer's has difference in the hippocampus, but also other places, but also in this major area where the fluid goes through, which we call a ventricle. So this is probably over 20 years old, this example, but this is really where the first evidence started, and it was at the Mayo Clinic. These days, we have more advanced software, and we can colour code them, and we can say, okay, all this green is the grey matter, the thinking cells. And then we have all the white matter. So you may have heard of all the grey matter, the white matter. White matter doesn't think. White matter communicates between grey cells, grey matter. And all the red is all the liquid. By the way, if we didn't have liquid, we'd give ourselves brain damage every time we walked because the brain moves with every heartbeat. And Sam is quite famous for developing a technique that actually shows that in real time. You see the brain moving, which is quite unique. And even shows more so that if we didn't have liquid every time we walked, our brain moves anyway and we just scrape because the inside of our skull is bone. And some of it, believe it or not, is a little bit spiky as well. So we need this liquid, it bathes, it suspends our brain. Okay, size isn't everything. So let's do another sort of MRI. It doesn't look at size. It just looks at things which should be there versus things which are there which should not be there. And this sort of scan shows those things as bright white. Bright white in areas where we know should be liquid is fine. But if bright white shows in areas which should be brain matter, that's not good. There's no functional brain there. So we call them white matter hyperintensities. If something is hyperintense, it means it's bright. If something is hypo-intense, it means it's black. See these little white things at the end? We call them caps. This is mild, moderate, severe white matter hyperintensities. And this commonly is seen in serial time points of people with Alzheimer's disease. The conundrum here is that it also happens in normal aging. So you need someone experienced, in other words, a radiologist, an experienced radiologist who has learnt during their registrar years, hopefully, that if you see this, doesn't mean it's dementia. How old's the person? It's not that hard to look at a scan and say this person is approximately this age group, but you can't generalise like that. So this can be another marker of either advanced aging. Why is it advanced? It might be dementia. It might be something else. So it's how often they're there, how bigger do they get, but it also correlates with age. But here's a second caveat. It also linked to depression. So people can have these white caps, but there are different types of white caps. If they look like white holes, we call it punctate. And people thought for decades, what are these things on MRI showing as these white punctate? It's called small vessel disease. Things are going wrong in the blood vessels. And as years went on and people followed them up, they thought, oh my goodness, this is not dementia. This is the vascular system breaking down, and we see it as depression. And it's called late onset depression, not dementia. But these things took decades to discover because you had to follow up these people. But now we colour code it all. So he's mild, moderate, severe. It's severe, but the person might be 100. So it's absolutely normal. So everything has to be taken within context. And you need a neuropsychological evaluation. We can get 2 scans, they might be a year apart, really doesn't matter, and you just overlay them on the computer and you say, show me in colour what wasn't there last time. We're just talking about these white dots, and now what is no longer there. And you think, how can it no longer be there? Is this person getting better? This is really good for treatment therapy. There's not many treatments for Alzheimer's, but you can treat things like Parkinson's at a certain level. So they might have had a plaque, multiple sclerosis plaque, which shows up as bright white, and then you treat them, and if you're successful, then these white matter plaques won't be there next time. So we can add, we can subtract, we can see change over time. Whew. Okay, so now we've looked at how big is something, how small is something. Okay, but now I just want to briefly talk about something I've been focusing on probably for 15 years now, and that's what about connectivity? The size of something tells us nothing about connection. How strong are these connections between our brains? Well, believe it or not, the connections between our brains is all about water flowing. If we heat up the water a little, we can follow the direction of the water. So think of grey matter and grey matter, the thinking cells. And now we've got the white matter connecting it. The white matter, let's think as a garden hose. Okay, the water's going back and forth. We'll just say, for the sake of argument, the water's going one way. Okay, it's really strong, the hose is really strong, there are no leaks. I'm going to give that a value of 1. But now, over time, my hose is getting a bit brittle. It's getting a few bumps in it. The water no longer is just going straight along. The value might be 0.8. Okay, now I've got the onset of neuroinflammation, which could be due to a number of disorders or environmental, or even we know now depression there's a class of it due to neuroinflammation. Our communication fibres, the white matter, become inflamed for whatever reason. And now our hose is a bit rippled and now the water's leaking a bit out the side. Well, now the value, let's say, is 0.5. So we can measure all that in real time in MRI. So as the water travels, traveling water we call diffusion. As the water diffuses, we call it diffusion MRI. And a direction, instead of a direction, let's get a bit mathematical, we'll call it a vector, or we'll call it a tensor. So in the literature, we call it diffusion tensor imaging, DTI. So the technique was developed in about 1988 by someone Sam actually worked with at Stanford. This guy's pretty famous. And then in 1991, another person came along and said, you know what, I can color code this. Everything right to left, I'm going to make red, up and down are blue, forward and back are going to be green. And all the colors in between are going to be the different angles. So this is where we're at now. So this was acquired on, not the one at Martai, but the exact same model of scanner. So this is actually my brain. And every voxel here is 1.2 millimeters wide, 1.2 millimeters higher, and 1.2 millimeters thick. You just think about that for a moment. Generally, we scan, for various reasons, 25.6 centimeters in all directions, because that's the average size of a head. And now try and imagine how many voxels at 1.2 millimeters would have to do. It's pretty remarkable. Maybe 5 years ago, to be really good, was about 3 millimeters at a time, which is still pretty amazing. Most people do 2 millimeters, but once you have what Sam called the Lamborghini of MRI, you can go down to 1.2 quite easily. You can even go sub 1 millimeter. So you're talking now in terms of hundreds of microns. And this doesn't take all day to do. This might take 10 or 15 minutes to do. So developed in 1991, expanded upon in colour in 1994, but no one could really do it because we really didn't have the MRIs that could push it that hard, and we definitely didn't have the computing power. But then in around 2000, new chips, Intel technology, it all just took off. Really expanded and more people started to use it. So we can see here the complexity of what's inside the brain. Remember, this is not so much grey matter. This is stripping it away and saying what's underneath the grey matter. Just the connections, none of this thinks, it all just communicates. We can go one step further. Through very clever software, we can see how dense these tracks are. And to do that, I'm going to take the 1.2 millimeters and I'm going to zoom in to 300 microns, 0.3 of a millimeter at a time. So here's my whole brain, 0.3 millimeter at a time. It's really quite extraordinary. And then I've just zoomed up on the part here which wraps around my neck called the brainstem like this. Just to show this is not a trick, it's not a painting, this is real. So red is right to left, green is front and back, blue is up and down. And all the other colours show the different angles of it, the different shades, the different hues. And every voxel, every 300 micron, we can measure the strength in this scale from 0 to 1. So why am I showing this? There's my brain, just take away half my brain and that's what the middle of the brain looks like. Because what we can do is look at the differences between people who have different forms of depression, different forms of dementia, different forms of schizophrenia, different forms of whatever you want, and compare them all. 0.3 millimeter at a time in this example, overlay them all and say where are the difference in the connectivity that might be markers of someone who has this disorder. We've got our DSM criteria, our famous Bible of psychiatry, and we've got a whole lot of psychiatric assessment, neuropsychological assessment. We've got MRI, how big is something, how many white dots are there, but what about their connectivity? We can put all this together and try and work out what is their real diagnosis. We can look at individual parts of the brain. So these are the parts that basically control my legs and my arms. So a lot of research has been done on this. So I can tell you this big bump here, which from this perspective looks like an inverted omega, is just my thumb. And as the fibers go down, they cross, which is why the right-hand side of the brain controls the left side of the body, and vice versa. And we can go through all these. I'll show you a quick video that I love showing. So it spins around. So we show this at a lot of conferences. And there it is at 300 micron resolution. You can go back and forth. You can look at any sort of coordinate you want in any direction. I'm sure you get the picture. Okay, but you can use MRI for more than how big is something, how many white dots are there, and what's the strength of this and this. When we use something in our brain, we're thinking hard, even when we're moving our thumb. We're using oxygen, we're using glucose. So what's going to happen? The brain goes, oh, you're using this oxygen, you're using this glucose, I need more. So you get this rush of these ingredients. We can measure that. So if I move my thumb in the MRI scanner and I'm saying to the scanner, I want to look at the oxygenation or the glucose changes, depend on which technology I'm using, just show me what happens when I move my thumb versus not moving my thumb. Where in the brain is this rush? And it'll say, okay, you just move your thumb, it's only happening here. And you can see here, it looks like an inverted omega. So there's the inverted omega. And you can see it from any perspective. Why would you even want to use something like this on the thumb? Well, it turns out it's probably the most widely used application of this technology we call functional MRI, because when someone undergoes brain surgery, the last thing you want to do is go near the area of the brain responsible for movement. They might have a brain tumour, for example. You want to take out that tumour. You want to take out all the tumour, but you don't want them walking out not being able to walk properly. So you put them in the scanner, move your thumb, move your toes. We get all these areas light up, and the neurosurgeons go, phew, I'm not going near that area. And they demarcate it, and it's all hooked up to computers, and some people use things called Da Vinci robots. So they're robots with 6 to 8 arms. They're the ones operating, going in, but it's actually the neurosurgeon in this sort of old gaming arcade with goggles on, controlling each of the arms, but it's for ultimate precision. So something like, say, the neurosurgeon's a little bit nervous for some reason or might slip up, those things are a thing of the past. The robot can do it. Not everyone has it, but this is a really good use of that technology. But this is more relevant to psychiatry. So the person's in the scanner, and we say, I want you to think of nothing. I want you to think of nothing for, say, 6 to 10 minutes, and you'll start daydreaming. And what the MR does, it says, okay, there's rushes to parts of the brain at the same time that are sort of linked. We call them phase linked. What parts of the brain are getting a rush at the same time when you're doing nothing? This is your default mode, we call it. It's the default way that you sort of think. It turns out there is a default mode network in the human brain. It's quite remarkable. And it shows up in very particular places. As the technology evolved, people started saying, okay, this is what's normal. Is it in everyone? How big is something? Where are the white dots? How strong are the connections? But what about the phase locking? How does this all work? It turns out that this is different in people with depression versus schizophrenia versus different forms of dementia. They're all the same places, but the amount of that rush is statistically different. So for example, what happens in dementia is you get usually a higher rush when you give someone a task in the scanner. So one of the beautiful tasks that Dr. Dudley quickly went by, but is a very clever test, I'll give you some numbers or some letters, say them to me backwards. It's not an easy task to do. We call it working memory, because first you've got to memorize it, which is hippocampus. Then you've got to put it in your little storage box, which might be about 10 seconds, which is here, called the frontal cortex. Then you've got to manipulate it, and then you've got to output it in your mind. So if your brain is not working the way it used to, then you've got to try harder. So even more blood rushes to those areas. It's a compensation technique. So a lot of people with early dementia are very good at hiding their symptoms. They just think harder, but you can't fool the scanner. So here's an example, as I was alluding to before, there is unfortunately, fortunately, at least we know there is a genetic link to a predisposition of Alzheimer's, and we call it ApoE. And these are the differences in a functional task showing the genetics of dementia. And again, I don't like showing new studies. I start at the beginning. Some of the real seminal articles that first pointed these out. Okay, so we looked at the size of the something, the white dots, the connectivity, the blood flow. Okay, we've got this person, they've got symptoms, but it really doesn't seem to be anything different. Okay, we have another bag. We've got some more tools in MR. It's endless. Let's look at the chemical makeup of some cells. It's very well known what sort of chemical is related to the strength of a connectivity, the amount that a neuron can pulse, all these sorts of things. We know the ingredients, we know what it feeds off. We also know there are ratios of these chemicals that should be in balance. So we look at the spectrum, and this is a spectrum. So we call it spectroscopy, which is basically a chemical analysis. So first we scan the person and we say on the scanner, I'm going to pick that area, that voxel, and just tell me the makeup of that area. And by looking at these are chemical peaks, but we know through a lot of research that a certain peak equals a certain chemical. And we know through a lot of research that the chemical should be at this level. And once we look at it, we go, oh, that peak looks a bit low, this one looks a bit high. You put it all together, you say, okay, well, if there's something like we call NAA, which is a marker of integrity of these connections, if that's low, okay, it's got something to do with connectivity, for example. And by the way, the MATI scanner can do all of this. Not all scanners can do all of this. Okay, so all that looks fine, and we still haven't got anything. What's going on? Let's use another tool. Let's look at the speed of the blood flow. So the blood comes from the heart, and at about this level, we're going to go with the MR. We're going to magnetically tag it, and we're going to follow it into the brain. How long does it take? So we call it arterial spin labeling. These water molecules, or the blood, is spinning, and we're tagging it as it goes up. So it's spin labeling. Before this came along, you'd have to inject a contrast which changes the way that the MR picks up this signal. You don't always want contrast. Some people feel a bit warm, can take a while to get out of your body, yada yada. So we call this contrast without contrast. And we look at cerebral blood flow with it without having to inject anything. So here's a bit of an extreme example, but someone has had a stroke, they've had epilepsy. The blood flow is different to the part of the brain that seems to be affected, but 6 days later, it's recovering. So on the scan, the structure would have looked very different. There's so much different. How are you supposed to tell which area is mostly affected? Which area is actually recovered? This is a really nice way to do it. And of course, it turns out that there are differences between schizophrenia, types of depression, and dementia in the rate of their cerebral blood flow that you can pick up via this technique. So I'll quickly move on because we must be near the end. Oops. So one of the areas I published a lot on is depression after head injury. So in the early days, no one had published on this. People have head injury, a lot of people get depression. In fact, the literature says up to 77% of people who have head injury will develop depression and they won't even know it. These days, all the focus, the media focus is on CTE. But I won't go into that. People becoming suicidal, these, the famous stories about gridiron players becoming aggressive, having symptoms of dementia years after they've had, say, 50 severe head injuries. It's along that path. Why? Because when you have a head injury, you go like this and like this. So your head at the front, this is personality, emotion, parts of memory. Hit the back, this is more than just balance. We used to think it was balance, it's a lot more than that now. Cerebellum is Latin for little brain. This little area here is 10% of the grey matter in our brain, 10% of our thinking capacity in this little thing. So these are the two areas majorly affected. So we should look at the bone, because if we can see a crack in the bone somewhere, we know the area of the brain that's going to be affected. This is pretty good. People usually have a CT, which is radiation. So it's an X-ray done over and over as it goes around you. We can do this with MRI now, believe it or not. This is something even a few years ago people said, you cannot see bone with MRI. It's just not possible. Well, it turns out they were wrong. So this is CT, this is MRI. And you can clearly see the bone in that. CT, MRI. You can clearly see bone. So now we can use MRI, no radiation. We see a little crack after a head injury, bang, that's the area we're going to focus on. I'll move along really quickly. So in schizophrenia, sometimes it is obvious, just like in some cases of dementia, where volume is important. So it turns out that most of these areas I'm highlighting here, so it's a very frontal disorder, but it's not only frontal. And again, these are some examples that was size. This is blood flow, the functional MRI. You think a thought, which areas are you using most? Oh, replenish me with the oxygen. But there's differences between people who have schizophrenia and don't have schizophrenia. So it's the same areas utilised, they just utilise it differently. What about schizophrenia with people who hear voices? Yeah? If we're right-handed, receptive language is on the left. And there's a little bump here. And this is what we call auditory cortex. It's a little bump, a gyrus, discovered by this guy, Herschel. So it's Herschel's gyrus. Okay. We do functional MRI while someone who's hearing a voice, boom, it lights up. It's overactive. If you look at the volume of it, it's not just a bump, it's up to 4 bumps. It's absolutely overactive. So the diagnosis is, okay, well, I can see why you hear voices now. There are medication techniques, there are brain stimulation techniques, but diagnosis can be pretty important. There's also another part that is the last part of the brain to grow. So generally speaking, the brain stops growing and it's mature at 25. Why do teenagers, why are they all over the place? Because the brain is still wiring. The brain is there, but it hasn't been pruned. Believe it or not, we start off with a whole lot, but you've got to prune it, and that's all based on experience. The only part of the brain that keeps developing until you're roughly 45, believe it or not, is this massive piece that goes right from the front all the way to the back. And it's called the cingulate gyrus. And it happens to be that this little front bit is implicated in schizophrenia. There are communication fibres that go from the bottom all the way up to this area. Personality, further thought, we call executive functioning. It's all connected. So what's the point there? Nothing works in isolation. Everything is a network in the brain. Lastly, hippocampus, because this has always been my passion. People are always looking at the hippocampus, memory, blah, 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 findings weren't so clear. And about 20 years ago, I started saying to these specialists, why aren't you looking at the very back part of the hippocampus? We call it tail. You can't see it on MRI. Well, maybe you can. I was really fortunate to be working with a neurologist at the time, and with her help and people from the pathology department at ANU, we worked out on a brain scan where you can see the tail. Lo and behold, the area that differed in all these disorders, no exception, was the hippocampal tail. This tiny thing right at the back. Even separating people who responded to medication versus did not respond to medication. Unbelievable. Right at the back of the hippocampus. These are just, I'm really proud of these publications, some of the ones that I'd publish just more to prove a point. Never say to someone it's wrong. You can't do it. It just gives you more impetus to do it. So what is it? It's this little part right at the back. It's about the tail, believe it or not. So what is it about the tail? Well, here we go. What's the one thing that depression, schizophrenia, and dementia have in common, neuropsychologically, cognitively, beyond anything, believe it or not? Navigation. Before someone with emerging dementia starts forgetting things, forgetting names, they forget places. Navigation. Where did I live again? How did I go there? They might still remember your name, but the navigation is the first thing to go. You test someone with diagnosed major depression, their navigation will not be very good at all. And the same thing for diagnosed schizophrenia. They all have this in common. And where is it located? Only in the hippocampal tail. So I went back in the literature and found this magnificent series of studies, and they're called the London Taxi Studies. Here we go. So Professor Eleanor Maguire from the UK in the 90s said, you know what? I think the navigation might be in the posterior tail because a study from the 70s said in mice it might be there. So she got many London cab drivers, experienced and not experienced, and then compared them. And where was the difference? Hippocampal tail. And then in 2014, 3 people were awarded the Nobel Prize for this same thing. So we call it the human GPS. And where are these cells? In the posterior tail hippocampus. These are the same area that many psychiatric disorders have in common. The size of other things, they're not really comparable between disorders. The connectivity is all over the place, but they're not the same between disorders. Blood flow, blah, blah, blah. The only thing they have in common is the posterior, the back of the hippocampus. So I wrote one of my crazy articles a few years ago trying to put it all together, and this was my model in a nutshell of everything that I'd learned in 20 years, is that there's a direct relationship to navigation of the hippocampus, and it's always smaller in psychiatric illness. So put 2 and 2 together, this is what they all have in common. Went one step further, this is all over the years, and I said, why measure the whole hippocampus? What dementia, depression, schizophrenia all have in common is that they have smaller hippocampi. But why are we just measuring the whole thing? Let's just look at what is not there. And if it's not there, then it's black, then it's liquid. So let's measure these little holes inside the hippocampus, which are then called hippocampal sulcal cavities. Lo and behold, dementia, schizophrenia, depression, hypertension, smoking. They didn't show differences in any of these other types of MRI, except for these little holes. The point is, you don't need to look at the big things. You can look at the small things. It can be just as helpful. Big picture, how does it all come together? Nearly 100 years ago, there was a person named Papez, and he hypothesized based on post-mortem studies that there's a circuit in the brain that brings all of this together. The frontal, the part of this little bit of the cingulate, the hippocampus, and he called it the Papez circuit. Believe it or not, this is what psychiatric disorders have in common. The majority of psychiatric disorders have an affected Papez circuit. We can put it all together and we can say, let's say there's 1,000 points around the brain, we'll call them nodes, and now put all that information together, the size of stuff from MR, the white dots, the blood flow, the strength of the connectivity. We're going to put it all into one equation and now show me how strong everything is and color code it and then give me a number for each of those thousands of connections. How does it connect? And we call it a connectome. This is the latest and greatest in MRI, in research of all sorts of disorders. What is the connectome? So I thought, okay, I'm pretty confident I'm onto something here with the hippocampus. So we put someone in the scanner and scanned them for 1.5 hours instead of 10 minutes. So we get more information than anyone has ever gotten on the connectivity. We had a really good subject. He was a semi-professional triathlon. So he could stay still, breathe really nicely. And lo and behold, these are all the connections of the hippocampus in the human brain. And mostly, it is the Pepez circuit. So if you affect the back of the hippocampus, you're affecting the entire Pepez circuit. All the reward, the hard thinking, all of it comes to the back part of the hippocampus. So the take-home message is there are similar regions implicated, but to different extents, and they change differently over time. The biggest take-home message, no part of the brain works in isolation. Everything is a network, just like those connectomes show. I hope I haven't bored you, but this is what I've learned in 25 years, basically, trying to put it all into a nutshell.
So I just want to thank you, Jerome, for that just really clear and accessible talk. And I just know each one of those slides, Jerome could talk to for probably maybe 1 week just for one of those slides. So well done for getting it, everything in a nutshell. And just that, neat how you tied all those years of experience and work all into, to help us understand more about the brain and several. It actually has applications for so many different applications, ranging from neuropsychiatric disorders through to depression and dementia and stuff. It's really wonderful, impressive work. Just because of the time, what I'm going to do is that if you've got any questions for Jerome, please come again to our tent tomorrow. Jerome's going to be there all day, right, Jerome?
Oh, yeah.
Except for a few breaks. We'll let him go for a few breaks. But and he's actually been to our local store to buy the last pair of gumboots. So he and Sir Richard Paul, who's also coming with his gumboots, they'll be hanging out together along with Dr. Dudley in the tent. So definitely come along and have a chat. Yes, Sir Richard will be there too. And we've got Miriam Scudding as well, Associate Professor Miriam Scudding, who's just arrived, who will also be there with some 3D printed brains that she's got in her bag right there. So we've got so much exciting stuff. So thanks again, Jerome, and we'll move on to Danielle, and any questions will come tomorrow.